Okay, thank you very much for the organizers for asking me to talk about clinical genomics. And I would like to invite you to take a step back. I only have a couple of slides on CLL in this session, so in my talk. So this is really about how we can potentially implement clinical genomics at scale. And essentially improve diagnostics, not just for patients going into clinical trials and not just in CLL, but at on a much broader level. I should say that I'm not employed by Genomics England, but I am leading the Genomics England Clinical Interpretation Partnership for hemato-oncological diseases. So progress in medicine really depends on precise diagnosis, and I'm sure everybody in the audience is convinced by this, and we've seen so much data at this meeting about both MRD monitoring and also genomic and otherwise stratification strategies. So I just want to illustrate this from a death registry of the 17th century, where people have tried to start to classify a disease by phenotypes and the way the disease presents. So bitten by a mad doggy, that's a sort of the causal effect. Purple and spotted fever. This is an example of description. Really suddenly, the way how people die. When it got really bad, it's a British registry, of course, so it was French, and it was called the French pox. So you can see that over the years we have hardly improved on classifying disease, but clearly we want to get to a mechanistic classification, molecular classification of cancers. And so that's increasingly relevant in diagnosis and prognosis in our world of hematology. The World Health Organization has revised the classifications in 2016 for myeloid and lymphoid diseases. And we are aware of this, and it was basically primarily because of all the advances in genomics sequencing. In the NHS here in the UK, we've recently reviewed the requirements for genetic testing in cancer, and we came up with 605 individual tests that had to be performed across hematological diseases and cancer, of which 177 are for hematological cancers. So a huge number of tests that have to be done and that are now nationally prescribed in the new cancer test directory. And we know in CLL that we are also using and trying to use markers for risk stratification. So what are the roadblocks to realizing the potential of precision medicine and precision diagnostics? From my perspective, within oncology, there's a lack of evidence of clinical utility of biomarkers, and we're all struggling with this concept that we want to use biomarkers, but then we do retrospective analysis and prospective analysis, but actually when it comes to using them, the field has already moved on. A lot of the problems are relating to the positive predictive value of the tests, because yes, they might predict outcome in a group of patients, but they might not be good enough to actually predict outcome of individual patients. The immunoglobulin status, for example, is a good example where we know maybe 50% of people are cured after chemoimmunotherapy, but it's not 100% of people, it's not 90% of people. So the positive predictive value is a problem. And that's directly linked to precision. If we had a very precise biomarker, then this number would go up to much higher levels. Measures of utility, so how do we demonstrate that these biomarkers are really useful? Is it that because we can risk stratify, is that useful enough? Do we have to show that we're improving overall survival by using a biomarker? Of course, that's directly linked to the efficacy of the subsequent therapy. Do patients get increased access to therapy, be it through clinical trials or early access programs or the Cancer Drug Fund, like here in the UK? Or is it good enough if we can show that we are saving cost by using biomarkers? And then there's a whole range of problems to do with the quality of access to testing. If a patient is in an academic center, they may have a lot of they may have access to a lot of tests. If they are somewhere in a smaller hospital, they might not have any access to testing. And that's directly linked to training skill, know-how of laboratories, but also to costs and centralization and quality control, ultimately, of clinical diagnostics. So there's many concerns and many roadblocks to realizing precision diagnostics. And I think the public, patients, and also politicians have realized that there are roadblocks to implementation of precision diagnostics, and that's why clinical genomics programs across the world have now sprung out of almost out of nowhere. And this is a summary recently published at the beginning of this year of the current programs. There have actually since then been more programs. Ireland has started off Genomics Ireland, and various other initiatives have started. So what is their aim? It's essentially generally to increase the understanding of genetics and biology of disease. Their promise, in particular in rare disease, that taken together, rare disease is a common disease, so in particular in these cases that they would stop the diagnostic or shorten the diagnostic odyssey. In cancer, we think that they will lead to comprehensive detection of genetic mutations that might actually lead to improved and more precise risk stratification. But there are other more political reasons: wealth creation, investment into biotechnology, capacity building, training of bioinformaticians, and then also the modernization and rationalization of health services, because the single test that we do and offer in the healthcare systems are actually unaffordable because they cost a lot of money when you take them together. So the scope of national genomics programs go far beyond CLL, they go far beyond cancer, they go into population genetics. Successful programs are programs that, for example, have started to use whole genome sequencing in pediatric ITU. They also include population screening programs, and more recently they include looking at polygenic risk scores. So trying to identify patients at risk of obesity, diabetes, cardiovascular disease, hypertension by interrogating the genome. And then they go into precision medicine, pharmacogenomics, and infectious agents and sequencing of mitochondrial and viruses. Generally, the operations of these clinical genomic centers are very similar. There's a spread of various numbers of genomic medicine centers, and then sequencing is performed in a central sequencing facility, both for research purposes and also for clinical diagnostics. There's programs around data sharing ongoing, and the data is being fed back to genomics tumor boards or other genomics boards, consumers and patients. So what's going on in individual countries? I just picked arbitrarily a few, and I really can't go into the details in the interest of time. France announced France Médecine Génomique in 2025. This in 2015, and it runs until 2025. The plan is to open 21 centers where samples will undergo targeted sequencing, with 2 centers in the south and in the north of France where whole genome sequencing would be performed. 2 centers have now opened, Segoya and Oragen. And the platforms are being developed similar to the general outline that I've given at the beginning. And there's also obviously a research arm to it. I think Sweden are very well advanced. They have now 7 centers, and Richard Rosenquist, I think, has already spoken a lot about this. This kind of grew out of a research lab, a SciLife lab, and has now branched into diagnostics. For our purposes, a targeted lymphoid panel is planned to go live in 2020. In Germany has lagged behind with this. There's a big commercial laboratory headed up by Thorsten Haferlach in Munich for hematology, and he has now finished sequencing 5,000 genomes of patients with hematological malignancies. And this data is eagerly awaited, and I've heard that very soon Germany will actually start a whole genome sequencing program that has not been officially announced yet. In the US, there have been multiple attempts at getting this off the ground, and there are various initiatives, both for patients and people who are healthy, who want to undergo sequencing of their genome, and then also initiatives within the private sector around private insurance companies setting up precision genomics facilities, and then the basket studies that we all know, NCRI Match, that is particularly targeting patients with oncological diseases and doesn't really recruit hematological diseases. The EU has recently announced that EU countries will cooperate in linking genomic databases across borders. This is also linked with funding for additional genomes, but it's primarily around federating genomic information and also healthcare information across borders. That's now really too political, so I won't go into this any further. I just want to give you a brief update on the 100,000 Genome Project that I've been involved with over the last 5 years. We've now, the recruitment into the project finished in January of this year. Over 100,000 samples have been genomes have been collected and sequenced, and about half of those results, about 56,622 exactly, have been fed back to clinicians and to patients. The spread across the cancers is shown in this pie chart, and you can see that the hematological malignancies made up a small proportion of the overall tumors sequenced, about 1,200 patients. In the hematology main program, we have again a large spread of different diseases, primarily ALL, AML, CLL, and multiple myeloma. You've seen some of the CLL data and the beginning of an analysis in an earlier presentation. I just want to highlight here that we have started to feed live data from the NHS and to link this up. So not just clinical trial data, which is what the CLL pilot was, but also NHS data. So we know, for example, the age of these patients. We know the ICD-10 code of their underlying diagnosis. We know the top 20 drugs that they were treated with, and we also know what type of analyses were performed. So many of these patients were in studies, not just the FLAIR trial for CLL, but also UK ALL11 study and AML19 studies. We have got hospital episodes and other hospital data now becoming available. So in blue you can see the non-cancer treatment and the hospital episodes, and in red you see cancer treatments. And this is essentially, as you see, as patients are being diagnosed with cancer and then undergo whole genome sequencing, you can see from 1999 all the way up to the sequencing program how many times they've been up to the hospital or up to see their GP. This has been linked to a national quality insurance scheme, primarily around the genomes, not so much around the clinical data as yet. And our national external quality assurance scheme has been enlarged to include a big branch on genomics quality assessment. And we're basically submit samples, DNA samples, fresh frozen samples, paraffin-embedded samples. We can then submit targeted sequencing, whole genome sequences in various different shapes or forms, from FASTQ files to VCF files, to essentially make sure that our labs are producing valid data. The NEQUA scheme is running, is open to anybody in the world. And with the exception of Africa, you can see that we have got participation of literally everybody. Specifically for cancer, all the genomic medicine centers have now got genomic tumor boards in place, and they essentially go through the genomic information, and they will then distill it and decide what information exactly has to be fed back to the general multidisciplinary team meetings. With respect to the research arm, this was all about diagnosis. With respect to the research arm, the clinical interpretation partnerships, of which there is 2 in hematology, 1 for constitutional diseases and 1 for the oncological diseases that I head up. The GSIPs were launched in 2014. We have now almost 400 academic institutions on board and over 3,000 researchers worldwide. And just to briefly illustrate again, for those who haven't looked at the abstract yet, so we have run the CLL pilot that predates this main program and was sort of a pilot to establish literally all the different aspects of the Genomics England program, from consenting patients all the way to interpretation of the results. And this was really because although there's whole genome data available in CLL, very little was available in the patients who need treatment. And no whole genome genome data was available from clinical trials. So we have now finished the sequencing of 488 patients with CLL in the pilot from 6 different clinical trials. And Katie went through this before, but we can basically, using very clever statistical tools, we have tried to combine all the different types of information that you can get from whole genome sequencing to come up with a prognostic and predictive model. And you see that this, at the moment, differentiates our 488 different patients according to progression-free survival into 4 different groups. And they do not really, they don't overlap with the existing single gene testing, whether it's TP53 testing or immunoglobulin testing. So we are beginning to see new prognostic and predictive groups emerging out of this analysis. So where are we going from here on the NHS side, so on the diagnostic side? The NHS has benefited from this to restructure the whole of genetic services in the UK, which has been extremely disruptive. However, the good news is that the NHS will offer whole genome sequencing as a routine diagnostic test for AML, ALL, all the childhood cancers and sarcomas, beginning in January of next year. They will also start to feed back results from pharmacogenomics and pharmacogenetics about toxicity, predicting SNPs of various shapes and forms. And they have been given money for 500,000 whole genomes over the next 5 years to continue this sequencing program in the NHS. A lot of this will go towards newborn, not newborn screening, but neonatal testing, increased pharmacogenomics, inherited disease and rare disease. And then as far as the oncology is concerned, we are very optimistic that a lot of the whole genome sequencing will be used to further characterize patients who are on expensive drugs. And that includes ibrutinib, venetoclax, CAR T cells, bone marrow transplantation in hematology. And it would hopefully also include a large proportion of our investigator-led clinical trials. So all of these patients will get whole genome sequencing done before they go onto these expensive drugs. So in conclusion, genomic testing at national scale is here to stay, not just in the UK, but in many other countries around the world. This is really to overcome the current reality when our precision diagnostics is very often only funded by private insurers, charities, pharma, research grants, institutional soft money, etc. We need to provide affordable and equitable access to precision diagnostics. That's one of the main arguments by politicians. We need to also ensure that big data is shared securely across population and national borders. And again, that's a political argument. And then in cancer, I think the clinical utility is really a big question. And so in the second phase of the Genomics England program for cancer, we are very proud that they will hopefully follow the example of the CLL pilot and focus on sequencing of patients recruited into clinical trials. And I will stop here, and I will thank the entire CLL pilot consortium. Thank you.
Thanks, Anna. We have time for a few questions. And one question that I would have is, it's a very impressive and worldwide program. As I understand you presenting this, are there currently ways that you're talking to each other through this worldwide consortium? Or?
Yes. So the consortium is not worldwide. There's essentially independent clinical genomics program that are funded through various sources, governments. A lot of it is private money, and research organizations. And we are, however, talking to each other, and we're trying to share as much information as we can and exchange experience. So there have been regular meetings. There's also the Global Alliance that is working primarily around harmonization of genomics analytical pipelines, data sharing strategies, and yeah. So there is a lot of communication happening.
Thank you very much. Thanks again.